So I welcome everyone to the meeting. There'll be a few folks, I think, coming in a little late from breakfast. I apologize for my voice. I think it's the first downside of coming back to a meeting, having not been to a meeting for 2 years. My voice is very raspy. And I was just talking to Mark Febbraio, and I think we've been just chatting so much in this dry air. So do apologize, and I might have to stop and get a bit of water. So today we're going to talk about extracellular vesicles, which is, I think, a really interesting topic. I'm going to go through some basics about extracellular vesicles and sort of set the stage for what we're going to talk about today. So if I could get the first slide, please. All right, so I'm going to be talking about the role of extracellular vesicles and the role in aging and exercise. I was going to mention a little bit about fatty liver, but I don't think I have time to get into that. But we'll touch on that a little bit more in the next talk that I'll be giving shortly after this. First of all, I just wanted to thank Dave Hood for organizing this. It's been a wonderful meeting. I think it's exciting for all of us to be back. It's been a long time since we've been in a meeting, and I'd like to thank him and all of you for coming here to attend this meeting. It's always a great meeting. It's been well over 2 decades since IBEC started, and it's been a delight to be to many of those meetings, and really appreciate the invitation to speak here today. So, of course, before I start, I just have to disclose that I am an employee of McMaster University. These are some of the folks that I've received remuneration for various tasks. And most importantly, I just want to declare that I did start a company called Exerkine Corporation, and we did have some interest in gene therapy using exosomes at one point. But you'll see today why that was absolutely slaughtered, and we went bankrupt. And good thing I have a very supportive wife, and we restarted the company as Stay Above Nutrition. So sitting in the audience and next to speak is Mark Febbraio, and I just want to thank Bente Peterson and Mark for introducing the concept of myokines. And we're all familiar with that story where they described IL-6 as the first myokine, but clearly, as you see in this adapted slide, there have been many myokines which have been discovered. And essentially what this concept refers to is that muscles can produce factors which are released into the interstitial fluid, eventually functioning almost as an endocrine signaling molecule to distant tissues. And that concept refers to muscle as being the generator. 15 years ago in my lab meeting, I said, you know, why don't we call them exerkines? Because they don't necessarily come from muscle. There may be feedback where liver could talk to muscle, fat could talk to muscle, fat could talk to liver, and vice versa. So the only conceptual difference is we refer to exerkines as the sum of all of the proteins in microRNA, and in this case, microvesicles and extracellular vesicles, which go up in response to exercise, which can mediate either signaling or other intercellular communication effects. So what are exosomes? Exosomes were first described in the early 1980s by a Canadian, Rose Johnston, who was investigating transferrin cycling. And she was the first to show that the transferrin binds to a transferrin receptor, is endocytosed, becomes part of an early endosome. And the endosomes can traffic in ways which we're still further defining into multivesicular endosomes, which can either go into the lysosome or be expelled as exosomes. We now know that there's communication between the endosome and mitochondria, and in fact, some of the mitochondrial turnover in mitophagy, we have some evidence that some of the mitochondrial DNA fragments can be spit out through exosomes. So it may be a form fruste which is involved in mitophagy, but that's a story yet to come. So we became interested in exosomes because it was clear that some of the canonical myokines, of course, we have to highlight the first one that Mark Febbraio and Bente Peterson pointed out, IL-6, have been found in public databases to be found within microvesicles or exosomes. So there are 2 public databases. One's Exocarta and microvesicles.org, where there's a compendium of various proteins which are reported to be contained within exosomes or microvesicles. So I did a scan of this in a review that I did, and just highlighting a few. One is IL-6 for logical reasons, and IL-15 we'll talk about. Bruce Spiegelman discovered also metronil, which has a role in fat metabolism. But not everything signals through exosomes. And so I just want to just highlight a little bit of data that we have on IL-15, which was a known myokine, but we did find some exerkine effects. And this was published a few years ago by one of my grad students, Justin Crane, in Aging Cell. And what we did is we took young individuals, less than 35, and older adults who were either top sport athletes or masters athletes, and compared them to sedentary individuals. And what we did is we did blood sampling, took the serum and applied it to dermal fibroblast to see if there was an effect of age, an effect of acute exercise, or an effect of training on mitochondrial biogenesis in those fibroblasts. So what we found is that it appeared that it was the post-exercise serum, especially in active individuals, which led to mitochondrial biogenesis in fibroblasts. So therefore, it appeared that in the serum, if we compared that fraction of serum to those that weren't stimulating mitochondrial biogenesis and did comparative analysis, we could come up with some of the factors which were mediating mitochondrial biogenesis. So that was the comparison. Clearly, it is in this fraction here where we expected those proteins to exist. So we did a multiplex analysis of 42 different cytokines and growth factors and came up with 2 targets that didn't have inflammatory effects, and these included IL-15 and transforming growth factor beta. So what we found is that these were the ones in the active individuals where there was a big increase compared to the sedentary individuals. So the assumption was one of these 2 is probably involved in mediating the benefits of mitochondrial enhancement. So what we did is we used neutralizing antibodies to TNF-beta, and you can see that we still had the stimulatory effect of the serum. So by pulling out TNF-beta, we didn't lose the stimulatory effect. However, when we pulled out IL-15, we lost the stimulatory effect. So this was evidence that IL-15 was at least one of the components necessary for the mitochondrial biogenesis. But to go further, what Justin did is we exercised animals 7 days a week for 30 days versus pulses of IL-15. So we used recombinant murine IL-15. And what we had to do with the IL-15 was to check with acute exercise what was the endogenous response so that we could mimic that by figuring out what concentration we had to give so that we could mimic this pulse. And it was an incredibly small amount, which is interesting because I think people who looked at transgenic overexpression or knockouts didn't really come to the same conclusion. But what we found is that if you tripled or quadrupled the amount that we were giving, you actually had an inhibitory effect on mitochondrial biogenesis. So it's a very small window. But what we found was that there was a stimulatory effect in the animals that was similar between the IL-15 and exercise for both skin and skeletal muscle. However, let's get back to exosomes and intracellular communication, because that was a naked protein that was injected. But are these carried in various vesicles? So what vesicles do we have? Well, there's a variety of things. Obviously, a cell, to put it into context of the size of, there's many sizes of cells, but this is an average size. And then cells can break down through a process called apoptosis and release apoptotic bodies into the interstitial fluid. There's also microvesicles, which can be released by cells, and exosomes. The problem when trying to separate these is you can see that there's an overlap between common things like viruses, bacteria, but the particle that we're most concerned about is the lipoproteins, because they're so abundant. And separating these lipoproteins from these other microvesicles remains a challenge, because many of the proteins or the lipids on the surface can cross-react with some of the things that we're using. So for people who use ExoQuick, essentially you're pulling down viruses, bacteria, lipoproteins, everything comes out with ExoQuick, which is really just polyethylene glycol. For any of you who've used that to isolate exosomes, you're pulling down all of these particles unless you do preparatory work. And we'll talk about that in just a minute. So first of all, before we even started to think about exosomes involved in the adaptation to exercise or with aging, we wanted to see if some of the machinery was in muscle. And this is some work that we've done just using antibodies. This is to TSG 101, one of the canonical markers. And then Alicia Salim, it was in my lab a few years ago, used immunogold. And this is IL-6 on an exosome just before it was being released. So it does show that in skeletal muscle, we do have some of the machinery, we do have exosomes, and some of the canonical myokines, IL-6 in this case, can be associated with exosomes. So as I mentioned, one of the challenges is isolation of exosomes. And I've already brought up this ExoQuick, which is really just polyethylene glycol precipitation, which is what the virologists were using in the 50s, and they knew that this could pull out viruses, but it also pulls out a whole number of particles. If you pull down with ExoQuick, you can see you get these clumps because there's just a mishmash of everything. But this may be adequate depending on what your question is. If you want to see if there is something specifically in an exosome or a microvesicle or an HDL particle, this would be an inadequate method because you're pulling down too much. So another way of doing isolation preparation is ultracentrifugation. And essentially, with various spins, you can spin down these particles. To get a little more specific, a classic technique is density gradient, things like sucrose gradient. Now keep in mind with this, of course, those of you who've done it, it's quite cumbersome, and you end up with a pretty low yield. So that's, I think, why a lot of people don't use it. You can use OptiPrep, and there's various other ways to do this, but you end up with quite a small yield, but a more pure fraction. We're going to talk a little bit about size exclusion chromatography. We think that's one of the best ways to prep, but there are downsides to this. One is the sample size that you get, and it certainly would not be scalable to a commercial use in the future if these were to be used commercially. And there are other things like tangential flow, lineal filtration, and one of the ways to pull out specific exosomes that have specific markers is to use an antibody bead method to pull them down with magnetic beads. So we're going to talk a little bit about the acute effect of exercise on microvesicles and exosomes, and just show you the preparation technique that we use. So there's a number of ways that you can isolate. You can get exosomes from serum. You can get them from platelet-free plasma or frozen. Just a word of caution, and this has been published over 10 years ago, that a study used a variety of different preservatives, and the only one that allowed for good extraction once it was frozen was sodium citrate. Now, acutely, if you prep your plasma and isolate your exosomes, EDTA is fine. Sodium citrate is, as I mentioned, very good. The one problem with serum is that serum, if you have a clotting accelerator, it takes half an hour. If you don't have a clotting accelerator, it's about an hour till it clots. During that time, the platelets are still there. And the platelets, unless you use something like Burfeldin A to limit the release of exosomes, they're going to be continually releasing into that environment. And so what you may see is almost a run-on from what had just happened. So from an exercise perspective, you may have a run-on from some of the particles that are there, unless you use an inhibitor like Burfeldin A, just for the grad students to think about some of these things. So with the size exclusion chromatography, what we do is a preparative step with low speed centrifugation to get rid of the large apoptotic bodies, higher speed centrifugation to get rid of microvesicles. Then we put it through size exclusion chromatography, and I'll show you that in a minute, and collect specific fractions. So this is work from Don Zudi, who did his master's in my lab, and we have a paper in revision right now, just looking at some of the different methods. So if you use some of the canonical markers, ALEX, TSG101, CD81, CD9, which are the canonical markers of exosomes, you can see if you just use those, things look great for just doing it straight out of plasma, ultracentrifugation, polyethylene glycol, or size exclusion chromatography. The issue comes in, however, is when you start looking at contaminants. So albumin, you can see the albumin enrichment in some of these methods. ApoA1, which is on the HDLs, you can see with all of the methods you do get some ApoA1, and that's even a problem with size exclusion chromatography. And then, of course, some of the other contaminants which one can see. So it depends on what your question is in terms of how pure you want your prep to be. And you may get the right answer from other methods, but just know what's coming down. And I think it's prudent now, especially given so much of the literature where people haven't been reporting what's the contamination, what method they've been using, and being aware of these things, what are you actually looking at? So with size exclusion chromatography, what we see is these nice cup-shaped exosomes, which have been well described, and this is what they should look like, these little cup-shaped exosomes, and generally their size is around 80 to around 130 nanometers. So looking at the acute effects of exercise, I'm just going to go through a few papers. There's a whole bunch of them, and Josh Netterveen, one of my postdocs, did a nice review, which I'll highlight in just a minute, published in Frontiers of Physiology, and this is one of the pictures from this. And right now, we really don't know much about the different types of exercise, HIIT versus moderate intensity continuous, weight training, et cetera. And it's a wide open area that I think needs further exploration, but we do have some insight as to what the influence of certain types of exercise are on the release of extracellular vesicles. So therefore, what happens to blood levels with exercise? So this is the first study that I'm aware of where people looked at acute exercise, and what they did was treadmill exercise and cyclergometer to exhaustion, young healthy males, and what they did is did a MV fraction, which was the 10,000 G pellet, and then they did a small EV and exosome fraction. So you can filter as well with a 200 micron filter to try and only have particles smaller than 200 microns, and then spin with ultracentrifugation. And they call that small EVs and exosomes. And I'll just highlight here, this is before exercise, this is immediately post-exercise, and then into recovery. And this is subject A, subject B. But if you look at the average, what they found was the total number of particles do go up when they looked at the small EVs and exosomes. But when they looked at the microvesicle fraction, the larger fraction, that didn't seem to go up. And I'll just very briefly mention this paper because my esteemed colleague, Dr. Mark Febbraio, is going to talk about this in just a minute. But it pretty much shows the same thing, and that is that with acute exercise, you can see here that there's an increase, and then with recovery, it comes back down. And they did proteomics. I won't get into it because I know Mark's going to talk about it, showing that there are a variety of proteins, even many of the glycolytic proteins, which can be contained within this. And again, deepest respect for Mark, fantastic scientist. The preparation, however, was probably not as pure as we would like to see for just exosomes. So again, science is real. It's just maybe that there are other fractions like microvesicles which contain some of these. And I think that question remains to be known. And I'd love to collaborate with Mark to see what's in our microvesicles. We've tried some proteomics with others, not as sophisticated as what you did, and the protein abundance was quite low, but it could be a technical fraction. We haven't published that yet. We have done some work with Louise Deldique, one of her graduate students. Joffrey came to work with us in the lab, and we used size exclusion chromatography that I just described. And bottom line here is that they're finding pretty much the same thing. The number of particles tend to go up and come back down. What's interesting, and I'm going to go through this in a minute, is when you look at the canonical markers, so things like TSG101, ALEX, and some of the tetraspanins, you can see that there's different patterns. So although the particles consistently seem to go up, I think what's in the particles is what's really changing. And that's where we really have to spend, I think, the next 5 to 10 years to really sort this out. What are these specific particles coming out, and are they enriched in TSG101, ALEX, et cetera? And I'll summarize that in just a minute. So again, this is some of Don's work in revision in Journal of Physiology. And what we did essentially was had 18 young, 18 old men and women. Half were sedentary, half were athletes. And we did a VO2 max test. And what we wanted to see was the effect of aging and the effect of acute exercise. And first of all, what I'll show you here, these are untrained young individuals showing very similar work to what Mark's shown and Ferber's group has shown, showing an increase with acute exercise and then coming back. So I think that's pretty canonical that when unaccustomed exercise is performed, you see an increase depending on what marker you're looking at. Consistently with ALEX, we see this increase. But something that I think needs further investigation, and hopefully some of the grad students in this room might do this, these are endurance-trained individuals, and you can see that they don't get that same response. So it almost makes me wonder is, could the acute effect in an untrained individual almost be a form fruste of cleaning out the cell, and it's more of a danger response, whereas those that are adapted over a period of time, we're not seeing that because there's no need to sort of prune your mitochondria and get rid of fragments with mitophagy and other things. And we see this also with some of the cytokines, that acute exercise in an untrained individual, you get an increase in the cytokines, but a blunting and adaptation once we become trained. And we saw that also with the old individuals. So again, with the top sport older individuals, we didn't see this increase. Now, one of the things that we did see, and this was the same with ALEX and TSG101. I won't show you the data because it's pretty much superimposed. This is ALEX, but TSG101 was the same. But what's interesting is when we looked at CD81 and CD9, and I think Aisha Salim, one of her students, was showing the same thing a few days ago on a poster, and that is with aging, what we see is that the CD81 and the CD9 exosome abundance is lower, and it's consistently lower. But we also don't see an acute increase when we use these markers, which is interesting. So it seems that the particles that are coming out are ALEX and TSG101, but not CD9 and CD81. But consistently, we can see that there's a lower abundance of CD81 and CD9. So it's nice to see that Aisha's group independently has shown something similar with this lower abundance with aging. But we'll explore that a bit more. So this is just data that we pulled out from a paper that's in press right now in FASEB Journal, where we're looking at acute exercise response with metabolomics compared to CSF. So we compared plasma to CSF. But what I did for this meeting is I pulled out the metabolites, which are reflective of exosomes. So these are some of the sphingomyelins, which are on the surface. So it's a different way of looking at the same question. Instead of particle analysis or Western blot to look at the canonical markers, we use metabolomics to look for the exosome markers. And just it doesn't matter what sphingomyelin species you look at, you can pretty much see that before exercise versus post, there's an increase, and then it comes back down in recovery. So I think that's a pretty consistent response now with multiple platforms of technology showing the same thing. So in summary, for the acute effective exercise, again, Josh did a nice job of reviewing every paper that's been published to date. What we can see is that with exercise, there is a stimulus for the release of small microvesicles and exosomes into the circulation. What we don't know now is exactly what's the proportion from different tissues like fat, muscle, versus blood cells, but I'll show you in just a minute. We have some of that answer, but these are released into circulation, and I think it depends on which marker you're looking at. In Josh's review, based on other work, he was saying that there's an increase with CD9 and CD81. We're not seeing that when we look at that, so I think that's still a little bit of a question, and this really needs to be better sorted out. But I think what we really need in the field is for very accurate description of what the isolation method was. So I'm just briefly going to show some other data that Mats Nilsson collected the original data on with some exercise training, and Dawn and Josh Netterbein have been analyzing this. So what our question was does home-based resistance training alter exosome markers and microRNA? So we had 32 older adults. We did 3 times a week training for 3 months with therabands and showed increases in all of the expected things like strength and whatnot. But what we wanted to see here is that if we isolated exosomes and small microvesicles from young versus old and the effective resistance training, could it bring the older adults back towards that of a young? And so what we can see here with a completely independent cohort, I'll just draw your attention here to what I was talking about in the CD9, CD81, again, consistently lower in the older individuals. Unfortunately, with resistance training, we didn't see a strong return to normal after resistance training with any of the canonical markers. However, when we used, when we looked at the microRNAs, what you can see is that pretty consistently across the board, when we look at some of the canonical microRNAs that are involved in muscle hypertrophy, these were uniformly lower in older adults and to a large extent rescued following resistance training. And this was at-home TheraBand training, pretty relevant when we were talking about all the folks at home with COVID. So this is in preparation now. We hope to publish this pretty soon. But again, what we did is I've done metabolic analysis with Bob Navio, and this paper's in preparation still, where we looked at the same groups, the young sedentary versus older adults. And again, this is a completely separate cohorts. This is the 3rd cohort, so we're sampling from different groups. And what we did was metabolomics with Bob Navio. This is the details, which I won't get into, of his setup. And what we found is that there's a big difference between older adults and younger individuals, but there's quite a profound shift. This is the entire metabolome shifting with exercise. So the older adults who are athletic had a much more youthful metabolome, but even pre-post exercise, there was an effect. And I want to show you this because it relates to the exosomes. So what did we find was the main pathway that was affected by aging? Well, one of them was exosomes were down. That was the 3rd most enriched pathway that we saw. So that is consistent with our work that I just showed you using a different analysis. But following 3 months of endurance training 3 times a week for 35 minutes at 60% of VO2 max, the #1 pathway that changed was sphingolipid metabolism. Again, suggesting that this is an important pathway, but I still think we're really early on in our understanding of this. And I'll just finish very quickly with why Exokine abandoned gene therapy. So I'm a metabolic genetics specialist. I treat children and adults with genetic mitochondrial disease, and you'll hear more about that in the next session. With this disorder, many of them are mitochondrial DNA mutations, things like MELAS syndrome, which we'll talk about later. The point is that if we were thinking of gene therapy, we would have to deliver wild-type mitochondrial DNA to all of the tissues in the human body. So we were thinking, well, this is cool. This exosome stuff is really exciting. Maybe what we should do is let's take cultured cells like mesenchymal stem cells, immature dendritic cells, and we can culture them. We can isolate the exosomes, and we can use various transfection reagents, electroporation, lipotransfection, et cetera, to get mRNA or mitochondrial DNA to go into these, and then we can have an enriched exosome that we can infuse into the animal, or in this case, the human. Or what about an allogenic bone marrow transplant? Because that's another way of thinking about it. If you've got a wild type, or a healthy human, I should say, do a bone marrow transplant, their bone marrow, if this is the case, should be producing mitochondrial DNA that's wild type, and it should cure the disease. However, I was slaughtered by one of my patients, which made us go bankrupt. So this was a fellow that I had seen. His mother died of MELAS syndrome, which causes strokes and dementia. And he was a runner his whole life. So we were amazed at the fact that this guy could run a 3:20 marathon, which isn't great, but for a kid with MELAS syndrome. So I biopsied him, and his enrichment was 75% mutant heteroplasmy. So he should have been deaf, blind, diabetic, probably had a stroke at this point, but he'd run his whole life. So Frank Booth, power of medicine. But the point was that with this individual, he came back a year later, and he had acute myelogenous leukemia. So he had a bone marrow transplant. And I was thinking, holy crap, this is pretty exciting, because he's doing the whole conceptualization of what we want to do. He now has a different mitochondrial haplogroup in his bone marrow. So he's got mesenchymal stem cells spitting out exosomes in their perfectly native environment. We don't have to isolate them, and and this guy also came off all of his immune suppression agents. So he's a perfect biorepository of a fresh mitochondrial haplogroup. So if the theory was true, he should then be starting to enrich his tissues with the haplogroup that was donated to him by the donor, and he should dilute down his MELAS heteroplasmy. The second thing that was interesting is one of my buddies' daughter, unfortunately, had severe illness, was just up the street here at Sick Kids, had a bone marrow transplant, but she's not world champion, but a Canadian champion in orienteering. So she's been training her whole life with her dad's bone marrow, which is pretty exciting, because I thought, aha, she could be another individual where we could see, does dad's bone marrow haplogroup, which is different because mom is the one who gave the mitochondrial DNA to her, would it shift in? So we've got ethics approval and analyzed both of these individuals. So with my colleague Beckham Sadakovic, what we did is that we isolated the mitochondrial DNA from the blood in these individuals. And what was interesting is that if you do an apoptotic body enriched fraction, which is just that 10,000 G or microvesicle enriched or exosome enriched, once we finally spin it down, what you can see is the read counts of the donor are predominant in the apoptotic bodies and in the microvesicles. So what does that tell us? It tells us that most of the apoptotic bodies and microvesicles that are in circulation in everyone in this room is coming from your blood, coming from your bone marrow. But when we go down to the exosome enriched, you can see how it shifts down. So about 50% once we get to the exosome enriched is coming from blood, the rest is coming from another tissue. Now we have methods with antibodies, we can see which tissue it's coming from, but that's interesting that if you just do a crude prep, a lot of this is coming from blood. But this is the key point here, and that is if we look at the conceptualization from the donor's mitochondrial haplogroup, is it shifting into tissues? And this is my patient with MELAS syndrome. And we couldn't even find it with high density reads. So I think we had like 10,000 read depth in our mitochondrial DNA. We couldn't find a single molecule from the donor had made it into the muscle. So essentially what this did, slaughtered our whole idea about gene delivery, and we went bankrupt and restarted again. So unfortunately, although the exosomes did have this mitochondrial donor DNA, almost nothing was transferred. We don't know what happened. Could there be a rejection of foreign exosomes? There's some theory that mitochondrial DNA that comes in can be almost rejected or foreign, almost a MHC-like. But we still don't know about other nucleic acids or proteins. I'm a little bit skeptical that that might work. The other thing that we started to see is that if we take, in this case, an immortalized dendritic cell line, we isolate exosomes, and then we inject them into a mouse, by the 4th dose, we have 100% lethality. So as a metabolic genetics specialist, I was thinking this is going to be a big thing to overcome before we'd ever get into clinic. The other thing is, again, thanks to Mark Febbraio, and Matthew Wood has also shown this, and we've shown this with a different method using luciferase, and that is that if you take exosomes and you inject them, where do they end up? They end up in the liver in the reticular endothelial system. So if I was smart, I would have actually taken fragments of DNA for COVID-19, and because it was going to the reticular endothelial system. And we saw this about 10 years ago, but I was too dumb to notice at the time, and therefore I'm not a billionaire and didn't come up with the COVID vaccine. But it's interesting that the delivery predominantly ends up in the liver, and Matthew Wood's work was very, very well done, which showed that almost nothing, and it's the same here, almost nothing goes into skeletal muscle or brain. And what's there is probably just lodged into blood vessels. There's no one who's shown that you can actually get it into the tissue and then it expresses in the tissue. However, I'll just finish with a word of caution. And I was mortified yesterday. For fun, I did a little search. And I realized on my search that there are exosome clinics. And a patient had come to me and say, Mark, I want to go to the US and get exosomes. I said, I'll give you a break. You can't frickin' infuse exosomes. You're going to die. But there are non-regulated by the FDA exosome clinics. And you guys can all Google this right now. For $10,000 US, anyone here can go down to the US with this. This is their picture, so that looks very professional. And you go down there and look at these exosomes. Don't those look beautiful? For only $10,000, their exosome treatment can help you to tissue repair, anti-inflammation, anti-aging. It's a cell-free regenerative treatment, will rapidly stimulate your natural healing mechanism, promote faster healing and recovery for only $10,000 US. I am mortified, because if these really are exosomes, which they're almost certainly not, it's probably sugar water, but if they were, by the 4th dose, what's going to happen? Lethality. So I'll conclude with, we don't think that this is a good idea. We do know that from other research, exosomes and small MVs are a very good way to do a liquid biopsy for biomarker discovery. We know that they're involved in paracrine signaling in several tissues, and we're going to hear some great data on that in just a minute. I think they're also a form first of cellular control. Certainly with acute exercise, they're probably a way to prune the mitochondrial tree and get rid of mitochondrial fragments and other proteins. We know that the exosomes are essential to life. Unfortunately, I treat children with a disease called Niemann-Pick C disease, which is a failure of exosome migration, and that leads to a fatal CNS disorder. But I really don't think they're a viable therapy for delivery, at least of mitochondrial DNA, and please, no one in this room get an infusion. So I'm going to finish with that. I'm a few minutes late, so I'm going to not take time for questions. Maybe at the end we can do a few, but I want to move on to Mark Febbraio's talk. Thanks. Thank you very much. Next we have Dr. Lee. Thank you.
So thank you for the invitation. I'm a gerontologist. My daytime, I do aging research and mitochondrial research. So it's really exciting for me to come to exercise meetings because my research has a lot of interactions with exercise, as you'll see today. So I didn't put up my disclosure slides. I am a shareholder and advisor to a company called Cobar, which develops mitochondrial-derived peptides for clinical use. Right, so I titled the talk Powerhouse, Slaughterhouse, and Speaker of the House because I think most of us know that the mitochondria are our powerhouse. It generates the great majority of ATP in our cells, hence the powerhouse. It also deals with cell death, and if you can extend it to immunity these days in killing organisms, I call it the slaughterhouse. These two are very well established, but what really fascinates me is in between giving life and taking life, mitochondria seem to be regulating a lot of these processes. So regulation and coordination absolutely require communication, and hence we are really interested in mitochondria as the speaker of the house. How does it decide policies in a cell when there's environmental changes, which happens every second of our life? So we like to ask this question from the very beginning, as in the beginning of time of eukaryotic existence, where we go back to the endosymbiotic theory that basically says a free living bacteria joins forces with our ancestral cell, and one very special moment It's given tenure to become an organelle, mitochondria. And this is believed to be the tipping point of evolution by many people. And we think that this being a very common thing, bacteria living in cells, shifting to this situation required a lot of communication, and they were able to do so because they were able to communicate and coordinate. And they probably didn't come up with a whole new language just to communicate. They probably used communication factors that they already knew that were embedded in their DNA, such as those that are used by bacteria today still, small peptides and molecules that are derived from genes in their genome to talk to their own kind in quorum sensing, or to be more antagonistic and kill other organisms, which we collect as antibiotics. And we think that these small peptides have been the foundation of not only the existence or the birth of eukaryotic cells, but also the evolutionary process of our complexity that we have today. So when it comes to the alphabets of mitochondrial language or mitochondrial communication, if you will, there are several that we know very well, and we've heard a lot of some of them in this meeting. Metabolites like reactive oxygen species, transient molecules like calcium, damage-associated molecular patterns like mitochondrial DNA that we heard about today, and then genes or proteins that are encoded in the nucleus that can migrate to the mitochondria and then translocate back to the nucleus to report back on changes in the environment. And we add now a more inherent signal, small peptides that are encoded in the DNA that have been derived from the original bacteria to be used to coordinate cell function, which we collectively refer to as mitochondrial-derived peptides, or MDPs for short. So there are 8 published MDPs, humanin, which was the first one that was discovered from an Alzheimer's patient's brain, and then Schlepp's 1 through 6, small humanin-like peptides, and then MOTS, and a couple more that will be published very soon. From Hasi Cohen's lab at USC, who's one of the leaders in the field of MDPs, and one from my lab that we are trying to get published. So I'm going to focus the talk on MOTS, one particular peptide. So we named it MOTS because we found it in the 12S ribosomal region of the mitochondrial DNA. So we named it mitochondrial open reading frame from the 12S ribosomal RNA. Type C was a third open reading frame in this locus. And it yields a very small 16 amino acid peptide. And we can check its mitochondrial origin at the DNA, RNA, and peptide level. So for example, at the DNA level, we can use a system called row 0, where you can selectively deplete mitochondrial DNA as a whole. And you lose the expression of mitochondrial encoded proteins that are very well known, like cytochrome oxidase 1 and 2, as well as MOTS, but non-nuclear encoded GAPDH. At the RNA level, we can use a compound known as actinonin that can, again, deplete mitochondrial RNA selectively. And then we lose expression of cytochrome oxidase 1 and MOTS, but non-nuclear encoded GAPDH. And more recently, we have data from mass spectrometry assays where we can confirm the peptide sequence and also identified some potential post-translational modification sites. So one of the first things that we did was, it's a small peptide, so it's unlikely to be an enzyme or some of a structural thing. It must have something to do with more rapid regulatory component was the initial hypothesis. So we tested many things. This is one example of an experiment we did early on. We said, well, maybe we give mice a metabolic stress because this is a mitochondrial peptide. If anything, it would have something to do with metabolism. So we, in a blinded way, we started to do a lot of experiments. This is one of them. So we're looking at mice fed a normal diet and a high-fat diet, and you're looking at the percent weight gain over the course of 8 weeks. On a normal diet, mice retain their body weight pretty well, and MOTS injections at 0.5 milligrams per kilogram intraperitoneally do not really do much to their body weight. High-fat diet makes mice obese, and we were able to see a significant reduction of this diet-induced obesity. So I'm not going to go into too much detail. This is all published stuff. But this led to MOTS being developed as a drug. We just finished a phase 1 clinical trial with fatty liver and NASH as indication in obesity. So it's really exciting to see your data, Mark, on that. So summary of the first paper was that MOTS activates AMP kinase, which is something that everybody here probably knows or heard of. to regulate cell metabolism, target the muscles, targets skeletal muscle to regulate the insulin resistance. And this was a lot of work done with Andrea, who is sitting back there, Andrea Hebner. And like I just showed you, we can prevent diet-induced obesity and insulin resistance, as well as age-dependent insulin resistance. So by the time this paper was published, there was a lot of discussion about MOTS-e being related to exercise. And this review called Exercise Pills, the starting line, put MOTS-e in the category of mitokines, mitochondrial cytokine, that targets skeletal muscle. The mechanism is through AMP kinase, and the ultimate goal is to maintain or promote metabolic homeostasis, which has now become a key concept for us. And we've heard of myokines today by Mark. And I'll show you the data later, but we think this might be a myomytokine to make things more complicated. All right, so we also saw a paper on that time of a mass spec-based method of detecting MOTS-e in plasma for doping control purposes. I didn't know why this was going on, but now I know that there have been lots of people self-medicating MOTS-e, and you can find them on YouTube, which is kind of interesting. There's a clinic above my dentist who actually charges a very ridiculous sum, like $10,000 for this treatment. Anyways, so we said, why don't we actually do the study and see if MOTS-e really is involved in exercise? So we teamed up with Troy Murray at the University of Auckland, and he had 10 very brave undergraduate students, all males. Unfortunately, we didn't have any female applicants at that time. And he put them on he put them on a stationary bike and collected their blood and muscle before and after and during exercise. And we saw a very significant increase in the level of MOTS-e expression, or the levels in the skeletal muscle with exercise. And this is the quantification of all the subjects. There were 10 subjects, and there's a very significant increase of MOTS-e in the skeletal muscle as well as in circulation. Which is a little bit different profile than the muscles. So we don't know which one is coming from where. Is this made from the muscles, or is this being taken up by circulating levels, or is this increased because the muscle is secreting them, perhaps through exosomes? There's a paper showing that MOTS-e is detected in exosomes, in neuronal exosomes. So we confirmed that there was, in humans at least, a connection between MOTS-e expression and exercise, which was very exciting, but we couldn't inject those students with MOTS-e, so the next best thing we did was to bring in mice. We tested inbred mice, outbred mice, young and old, and here I'm showing a study we did with young and middle-aged and old mice. So we took black 6 N mice, 12 months of age and 22 months of age, and put them on a treadmill. This is the percent remaining on the treadmill as a function of time, and as expected, the older mice don't run as well as the middle-aged mice. Then we injected these mice with MOTS-e for 2 weeks, again, intraperitoneally, and then we were able to see almost a doubling in their running capacity, and they definitely outran their middle-aged counterparts. And the middle-aged mice also benefited from MOTS-e. And there seems to be a ceiling effect of how much this can go. So at that point, we were asking, how is this happening? And we knew, like I mentioned, in the previous study that we published with Andrea, that MOTS-e can target the skeletal muscle and improve their insulin sensitivity. So this is a study where we took, this is black 6 mice, and this is a typo, this is J, not N. At 12 months of age, these mice already show signs of insulin resistance in the skeletal muscle. So we took them in a muscle strip test that Andrea did. She gave them all equal amounts of insulin and looked at glucose uptake. And clearly, the muscles from the older mice were much less able to take up the glucose. And 1 week of MOTS-e injections in the 12-month mice was able to partially reverse this age-dependent muscle insulin resistance. So we said, well, maybe there's something to do with muscle metabolism that allows them to run better, because they're definitely not bulking up or anything like that. So with Nick Graham at USC, we did a metabolomic study. So we pulled these mice, 22.5-month-old mice that were treated with MOTS-e for 2 weeks, put them on a treadmill, exercised for 30 minutes. We pulled them out and immediately collected their muscles and ran metabolomics. And we confirmed that there is a strong effect on glycolysis, the glucose metabolism, as well as amino acid metabolism. So there's a metabolic reprogramming that's going on, and we're trying to figure out how that, I'll show you a little bit more data later on how this integrates into the pathways. So there's a metabolic reprogramming in the muscles by MOTS-e. And we said, oh, well, maybe it's trying to maintain homeostasis. So you're exercising, exercise has all sorts of stress, oxidative stress, metabolic stress, heat stress, mechanical stress, and maybe they're trying to help them cope with the stress. The mitochondria is sensing these changes, and it's communicating back saying, we need to strengthen up. So one of the studies we did was kind of model that in C2C12 cells, and like Mark said, it's not the best muscle cell line, but this is the one we started off using. And we deprived them with glucose in serum to mimic a fasting condition. We call it energy deficit stress, and we cultured them 48 hours with and without MOTS-e and stained them with crystal violet, and there was a pretty dramatic difference in survival of these C2C12 myoblasts. These are not differentiated. And then we said, well, maybe they're just there and they're just breathing, barely surviving. But one of the definitions of life is that you have to be able to reproduce. And for myoblasts, that's very important. So we said, let's see if they can actually proliferate if we put them back into normal conditions. So we extended the energy deficit for 7 days with and without MOTS-e. And after many of the cells die, we gave them back full media to ask the question, can they come back? And the effect was pretty strong. MOTS-e treated cells were able to fully cover the plates or the wells in 2 days, whereas the controls were not able to do so. So it seems like they are reprogramming the cells to cope with whatever is given to them in a better way. So at that point, being gerontologists, we couldn't resist the temptation to look at aging because the effect on exercise was much stronger than we anticipated. And so instead of euthanizing all the mice, we quickly adjusted our IBEC protocol to look at longevity. It's definitely not designed with enough power to look at lifespan studies, but we were so tempted to do so. So at 24 months of age, after some time has passed, we put the mice on an intermittent treatment because you can't inject them every day until they die. So 3 times a week, intraperitoneally, and until up to 32 months of age, we see an increase trend, p-value of 0.05, just needed 1 more mouse. But there's a trend of significant increase in lifespan, about 6 to 7% in median and maximum lifespan. So we're almost funded to do a full lifespan study for both males and females, and hopefully that works out. So going back to the question, how does this happen in the cell? And while we spent a lot of time under the microscope, me and my postdoc, we recognized that there are these speckles in the nucleus that we found. These are HEC293 cells that are just happy in normal media, if you can call them happy. And we thought these were artifacts, but then with this whole idea of adaptive homeostasis, we said, well, maybe it actually has a condition where it likes to go into the nucleus, like a lot of these stress proteins. And so we give them a lot of different kind of stress. Here I'm showing only glucose restriction. So within 30 minutes of withdrawing glucose down to fasting level, so that's 50 milligrams per deciliter, there's a rush of MOTS-e that goes into the nucleus, indicating that there's a nuclear interaction, something to do with perhaps gene expression. And this translocation into the nucleus requires AMP kinase because either if we pharmacologically inhibit or using genetic interventions like siRNA to get rid of the alpha subunit of AMP kinase, this increase in the nuclear compartment of MOTS-e after glucose restriction that we see is lost without AMP kinase. And I'm not showing it here, but AMP kinase activation itself is sufficient to trigger translocation into the nucleus. So there's a coordination between a nuclear-encoded energy sensor and a mitochondrial-encoded energy communicating factor. So, so all of this data I showed you is this is all endogenous levels of MOTS-e, naturally produced. We also looked at what happens to peptides that we give them, to give to the cells as a treatment. So we used a FITC, a fluorophore-labeled MOTS-e, and immediately after treating, we take them into the microscope, and we see small granules or signals inside the cell that's not in the nucleus. And then within 30 minutes, we see a pretty pretty strong presence in the nucleus. And then with time, it diminishes and gets cleared out. So in collaboration with Berenice Ben-Ayoun at USC, we did some genomic studies asking the question, how does this regulate genes? I'm not showing the data here, but in a previous paper, we demonstrated how MOTS can go into the nucleus, interact with transcription factors, and regulate certain genes. So this was building on that study, and we were just asking what genes are involved here. So what we did was we took the muscles from the old mice that were trained or not trained, and then looked at differential gene expression. We also looked at the C2C12 myoblasts with and without metabolic stress, and looked at the differentially regulated genes by MOTS, and then we overlaid those two differentially regulated genes to see if there was an overlap. And many of the significant categories, biological categories that changed had to do with protein regulation and protein metabolism, as well as oxidative stress, mitochondria metabolism, which we expected. So this was kind of unexpected for us. So we did an analysis called CHIA, where you look at transcription factors that can, in part, explain all the genes, or many of the genes. And so, again, overlaying the two RNA-seq data, we found one gene that was highly expressed, well, not expressed, highly likely to be responsible for the genes in both of the conditions or the models, and that was HSF1, H-cell factor 1. So we took it out. So this is metabolic stress again. This is controls. With and without MOTS, the ones treated with MOTS fare better under metabolic stress, but if we remove HSF1, the effect of MOTS is reversed. So indicating that HSF1 is indeed a critical transcription factor to mediate the effects of MOTS, at least in the muscles. So on a parallel line, I just wanted to touch a little bit on this because I wanted to bring the more intra-organelle, in addition to the intra-organelle actions of MOTS. We were collaborating with Min-Sung Kim and Min-Ho Seong in South Korea, and there was a model that that Min-Ho Seong developed. It's called CRIF1 knockout mice in the brain. Basically, it's a mito-ribo proteostatic stress model. It causes protein imbalance between the mitonuclear compartments. And we were looking to see how MOTS modulates this. But the long story short, from the context of exercise, we see that the working model is that the IL-6 exerkine that we've heard today goes into the brain and targets the hypothalamus, specifically in a region called the arcuate nucleus, where a lot of these POMC neurons are, and can target the POMC neurons to start expressing proteins like endorphin and MSH. But anyways, here in this process, we see MOTS being heavily involved in mediating these effects. MOTS can actually interact with STAT3 in the binding regions in the promoter of the POMC gene and enhance the expression of POMC. So here are some of the data. This is just regular exercise. 2 weeks of moderate exercise on a treadmill, 18 meters per minute for about an hour for 2 weeks. And then we looked at the expression of MOTS in red in the arcuate nucleus, and we see a pretty significant increase in expression, and many of them overlapping with the POMC neurons. And this is just a quantification. So the question was, well, it seems to be, MOTS seems to have a central role. And the question was, how does that translate into the periphery, the whole body? How does that hypothalamic muscle connection work? So we injected MOTS ICV, intracerebroventricularly, and we looked at, what we found was there wasn't a huge amount of difference other than fat, which was very interesting. What we started to see was a beiging of fat, a white adipose tissue, and the reduction of droplet size, lipid droplet size in the brown adipose tissue, indicating a shift in adipose metabolism. So I just wanted to touch base on this to build on the intra-organelle organ communication, that there's a muscle, that there's a brain, there's a liver, and they're all working together, and the mitochondria seems to be involved in all of these processes. So to summarize, I just wanted to emphasize that our genome is actually not only in the nucleus, it's in the nucleus and the mitochondria. In fact, the mitochondrial genome predates the nuclear genome and has been with us for the longest time. And these two actually work together in a cooperative fashion. So it's the cooperation of the co-evolved genomes. And there's a lot of proteins known that regulate the mitochondria, and we think we are identifying small peptides that can do the reverse. And this, the beauty of this is that it's not just intracellular communication. As we evolved, we became more than unicellular organisms, so the birth of endocrine regulation probably also has something to do with the mitochondrial way of communicating. These are found in high levels in in circulation, so the endocrine system is also consisted of nuclear and mitochondrial encoding factors. And exosomes, I think, are really an interesting topic, because one of the questions is how does MATI get into the cells? And exosomes can definitely explain how they can be taken up. And there are papers coming out showing that MATI is found in exosomes in the COVID-19 patients, pretty dramatic changes in neuronal exosomes in those that had psychosis or not in the brains. It's a paper from UCSF that came out a couple of months ago. Anyways, so I'd like to end my talk here and take some questions if there are any.
Thanks very much.
Thank you, fascinating talk. So given all these functions by MOTS-c, is there any evidence from JIVA studies that there are genetic variants associated with this disease?
Hassie Cohen does a lot of these genomic studies, and there's very clear ethnic variabilities. So there is a strong genetic background to this. For example, there is a specific haplogroup in the Japanese population that's linked to longevity that's not seen in other populations. But just to touch upon that, I think one of the underlying concepts of your question is that there is a compatibility issue between the nuclear genome and the mitochondrial genome, because when we breed, you can't really know what's coming from where. And so there are a lot of studies, like from many studies showing that if you move around the mitochondria in different nuclear backgrounds, the phenotype of the mouse is very, very strong, if you can even get them fertilized.
Yeah. But the question was, if you look at JIVA studies, so are there genetic association between SNPs and DNA variants in the MOTS-c gene and human disease?
Yes. So I'm blanking out here right now, but MELAS, there's connection with MELAS, and there's a strong connection to hearing loss that's connected to the ribosomal RNA gene.
Okay, thank you.
Does he want to go?
Oh, go ahead, Mark.
You go.
Oh, okay. Hi, so I want to ask about the tissue expression of MOTS-c. So you mentioned that it's expressed in many tissue and also present in circulation. So I was wondering whether during exercise, the MOTS-c expression was induced by a predominant organ like skeletal muscle, or some mitochondrial-rich organ that it goes into circulation and goes to some other organ like hypothalamus, or does it get expressed locally in hypothalamus?
Yeah, that's a great question. The same answer as Mark gave on the exosomes. We're not sure if it crosses the blood-brain barrier. There's one paper that says it doesn't. We're still trying to figure it out. So, yeah, it's an open question. It's an open question. Expression-wise, we see expression in a lot of tissues, especially the ones that are high in mitochondrial density. So the heart, the skeletal muscle, liver, those are the high. And the testis, for some reason, it's very high.
Okay, thank you.
There's I asked this question with reference to the doping. So there are a couple of genes, ACE2 is one, and actinin 3 is another, where variants determine athletic performance. Particularly with actinin 3, Kathy North has some very good data that if you have one variant or the other of this gene, you're predisposed to being endurance. Is there any evidence that with MOTS-C that there are any splice variants of the gene? And does it correlate to athletic performance? Because it should.
Right. So these are small open reading frames. There are no exons, and that's why
okay.
it was excluded from the first genome project.
Right.
It's not a gene, which is now being revived. There is a haplogroup, again, from a Japanese population that are highly enriched in high-performance athletes.
Yes, okay.
So there is a connection there. And the funny thing is that that mutation has a functional consequence, and it actually reduces insulin sensitivity a bit. So we don't know exactly how that's correlated, but there is.
Maybe you can just do an observational study where you just take biopsies from highly endurance-trained, elite athletes, and look at the abundance of MOTS-C in muscle to see if it's a marker of athletic performance.
Yeah, I'd love to do that. Yeah, I mean, open for collaborations. It's an excellent project.
Just one quick question. Is the expression of MOTS-C independent of mitochondrial DNA replication? So, I mean, do you have to have polymerase gamma and then you replicate the entire mitochondrial
That's
genome?
a good question. We're not sure right now. We know when MOTS-C gets induced, and that's typically linked to cell cycle. But then the mitochondrial replication is independent of the cell cycle. So we haven't really looked into mitochondrial division. We do know that TFAM is involved somehow in transporting MOTS-C transcripts. So there might be some connection to replication, but we're not entirely sure.
Great. Thanks so much. All right. Thank you. We're doing quite well on time. Now we have John McCarthy, who's done quite a bit of work in this area. So I'm very excited to hear this talk.
All right, so first I'd like to thank David for the invitation to come and share with you our work looking at intercellular communication. Before I get started, I should acknowledge for the people who don't know, all of this work was done in collaboration with Charlotte Peterson. So we agreed before, which she's just finding out about, if you have any issues with some of the data, she's going to field those questions. Any data that you love, I will field those questions. So, in trying to come up with a title, the ins and outs of exercise-induced skeletal muscle intercellular communication. So the ins, as you'll see, is going to refer to our work looking at satellite cells and their ability to communicate with other cells within the muscle. And the out is going to be some other work where we have shown that it looks like muscle releases exosomes that are preferentially taken up by white adipose tissue and promote lipolysis. Let's see. Okay, so to start the in, we go back to about 2014, and this is Chris Fry, who was a postdoc in the lab at the time. And he did a study where we had initially shown that in satellite cell depleted muscle, the increase in muscle weight in response to mechanical load induced by synergist ablation was pretty much the same. What he did is he took it out to 8 weeks, and what he found was in terms of muscle weight normalized to body weight, that there was a blunted growth response. And that was consistent with what he found at the single fiber level. So type 2A, it's hard to see, type 2A, 2X, I think it's 2X. The CSA was actually smaller. So something was happening between 2 and 6 or 8 weeks in which the muscle was unable to fully grow. So then he looked at the level of fibrosis, and what you can see here quite clearly is that by 4 weeks, there's a significant increase in the amount of fibrosis, and that is dramatically increased by 8 weeks. So somehow, the loss of satellite cells, the depletion of satellite cells is causing an increase in fibrosis to what we refer to as long-term mechanical overload. So, for the sake of time, I don't have the time to go into all the details, so I'm just going to summarize the findings, and this was in collaboration with Tyler Kirby, who was a graduate student at the time, worked with Chris, and both in vivo, in vitro evidence was consistent with a mechanism in which satellite cells become activated in response to mechanical loading. They release, I'll say, exosomes, and these exosomes in part contain miR-206, which turns out, at least in our hands, is the most abundant microRNA within these EVs, taken up by fibroblasts, and 206 represses expression of RBP1, which is a master regulator of collagen synthesis. And so in this way, satellite cells are helping to orchestrate or regulate remodeling of the extracellular matrix during hypertrophic growth, and that ensures that you get optimal growth. Now, in the absence of satellite cells, this whole mechanism is no longer operative, and that leads to an increase in fibrosis and the blunted hypertrophic growth that we noted at 8 weeks. However, one of the interesting findings was that if we did mechanical overload via synergist ablation and left the satellite cells there for the first week and then treated with tamoxifen and depleted, we were able to completely rescue the growth response as well as the fibrosis. So this suggested to us that satellite cells were doing something during that first week to, we'd like to think that they were communicating in this context with fibroblasts or maybe FAPs to regulate extracellular matrix remodeling. Okay, and so then following up on that work, Kevin Murek joined the lab. In his first project, which he might start twitching when I say NWASP, was to look at NWASP. And so we were trying to figure out a way in which we could prevent satellite cell fusion. We wanted to keep the satellite cells there, but we didn't want them to fuse, so that we could distinguish between fusogenic and non-fusogenic mechanisms. And so in doing that, he crossed the PAX7 Criara mouse to a reporter gene DT tomato. And so that's shown right here, satellite cells nicely labeled. He took that mouse and he did synergist ablation, mechanical overload for 7 days, and then digested the muscle, looked at it underneath the microscope, and as you would expect, saw satellite cells that were labeled, but he also saw these fibrogenic-like cells that also contained the DT tomato. When he isolated EVs from the media, he could see, in fact, that there was a lot of DT tomato mRNA. So at this point, that for us suggested that the DT tomato reporter gene was being packaged into EVs and could be delivered to other cell types. And so for anybody that uses reporter genes, especially in which you're using a tamoxifen-mediated, CRE-mediated recombination, this brings up a lot of issues, especially if you want to do some type of tracking. However, for us, it now represented a tool to be able to use DT tomato as a proxy for satellite cell communication. Okay. So this is just more in vitro evidence that Kevin had acquired, trying to really confirm, in fact, does satellite cell DT tomato mRNA and or protein get packaged into EVs, and is it capable of being delivered to other cells? So here is just showing that in the NWASP, when he knocked out NWASP within satellite cells, and of course he had the DT tomato reporter there to assess CRE-mediated recombination, how effective is he knocking down NWASP? You can see here after 7 days of mechanical overloading, we don't see any increase in myonuclear number, so that's indicative of satellite cell fusion. And in NWASP, at least at this point, we didn't see any satellite cell fusion. However, what he did see in the absence of that fusion is DT tomato within the myofibers, indicative of, at least our interpretation, that these exosomes containing DT tomato were fusing to myofibers. He also went and isolated primaries from those, isolated EVs and put those on C2, C12 myotubes. And you can see, I don't know how well you can see it here, but you can see that if they came from the, I think it was an PAC7 NWAS DT tomato mouse treated with tamoxifen, in fact, you could see that it was transferred into myotubes. So collectively, that data suggested or indicated to us that, in fact, we could use this to try to track or decipher satellite cell intercellular communication. So this was our first attempt at single-cell RNA sequencing using the PAC7 DT tomato mouse. And what you can see here is here are satellite cells, and as you would expect, they have high levels of DT tomato. However, you can detect DT tomato in these other cell types, such as FAPs, endothelial cells, macrophages, and of course, even though it was a single cell, I'm sure anybody else has experience with this, myonuclei always come through on the sort. And so here is the myonuclei, and you can see that there are ones that have a high level of DT tomato, and those are more than likely satellite cells that had fused to the myofiber. So this was some of the very first evidence that we had that, in fact, satellite cells, in response to mechanical overload, were communicating with these other cell types. So here is some more work by Kevin, and again, it's a little bit different. Here he had the mice were different. He had just the PAC7 DT tomato treated with tamoxifen, and then he also had our PAC7 DTA mouse treated with tamoxifen. So this gave us with or without satellite cells. And now we did single cell, and we're not so much looking for DT tomato, but we're trying to see what is the effect of when you lose satellite cells, and our thinking is that you're losing that communication. And so just to highlight some of the notable changes, you can see, and this is instead of 7 days, just 4 days. And the idea here is that by doing 4 days, we're avoiding satellite cell fusion. I mean, you do get some satellite cell fusion at 4 days, but it's minimal. And so what you can see in terms of FAPs, in the absence of satellite cells, there's significantly more FAPs. There's this fibrogenic population that is not a FAP, but it expresses fibrogenic genes. And then in terms of gene expression, consistent with what Tyler and Chris had shown, was that these FAPs have higher levels of collagen as well as an increase in cytokine expression. So our interpretation of that data is that in the absence of satellite cells, that communication is no longer there, and so that these other cell types, their activity or their abundance is being changed. So in a similar way in which Chris showed that these EVs are able to regulate expression of RRBP1, Kevin did the same thing, where what he first found out from the single cell was that WASP1 expression was higher, and WASP1 is important for, it needs to be downregulated in FAPs, and that is necessary for FAPs differentiation. And so he had 4 different conditions where he cultured MPCs with primary fibroblasts, or he took MPC conditioned media, or he had, it's hard to see, he isolated EVs from MPCs, or he just transfected in MIR206. And you can see under all 4 of these conditions that we're able to get a downregulation of WISP1. So the interpretation of that is that satellite cells become activated very much in the same way that we showed that MIR206 from exosomes is able to regulate RRBP1. In this case, now we have satellite cells communicating to FAPs, and they're necessary for the downregulation of WISP1, which allows for the FAPs to undergo proper differentiation. And this is probably one of the more important findings from that study, is, again, he repeated this, but instead of having satellite cells around for 7 days, now he did the mechanical overload via synergist ablation, kept satellite cells for 4 days, and then treated with tamoxifen to deplete satellite cells, and then allowed them to go out for 8 weeks. And under this is just data showing, in fact, that he completely blocked, depleted satellite cells, completely blocked myonuclear accretion. And I think the important point, or really from this experiment, was that the growth response was completely the same after 8 weeks. So whereas Chris and Tyler showed that as long as satellite cells were there for the first 7 days, you could rescue fibrosis, you could rescue hypertrophy. What Kevin found was now satellite cells, as long as they were there for the first 4 days, they created a permissive environment that allowed the muscle to fully grow. And again, so we look at that and interpret it as that satellite cells are communicating to these other cell types within the muscle. Okay, so that's all some of our published data, and so where are we going now? So really a big effort in our group is we'd really like to be able to tissue specifically label whether they're EVs from the myofiber or from satellite cells. And so Dr. Yang's lab at Tufts created this mouse in which they took the human CD63 infused GFP, and this is a Copilog GFP, which it's supposedly more stable and much brighter than enhanced GFP. And so he was kind enough to share this mouse with us, and of course we crossed it to our PAX7 GFP mouse with the intent of hopefully labeling satellite cell exosomes. And this is just some data just to convince you that, in fact, here is the endogenous CD63, and using this mouse, you can see that, in fact, you get very good co-localization. Here is just showing that you can track the CD63 GFP exosomes in the dendrite. So this is some work that is led by Yuan Wen, and more recently Ahmed Ismail joined the group. And so this is some of the earliest characterization of the PAX7 CD63 GFP mouse. Here is 1 week post-tamoxifen. Here is 4 weeks. You can see quite a bit of GFP. Here is a higher mag image of 1 week. And you can see here is a satellite cell and some GFP associated with it. However, there's other satellite cells in which we don't see any GFP. Here is from 4 weeks post-tamoxifen, and for us, this is actually really quite exciting. So here's a satellite cell. You can see some GFP here. Here's a merged image. So this is dystrophin. Here's a satellite cell, and more distal from the satellite cell, there is GFP. Here is a couple satellite cells, and you can see here in the merged image, there's a satellite cell, and GFP is throughout this neighboring myofiber. So we look at that, and again, this is all under resting conditions, right? So we just treated the mice with tamoxifen and waited a month. And we interpret this data, though it's very preliminary, that it appears to us, and kind of consistent with what Kevin showed with the PAX7 DTA, that it does appear that satellite cells are actively communicating even under resting conditions. Okay, so now I'd like to move on to the out. And here we kind of take a trip down memory lane. So unfortunately, Karen isn't here today, but this was the very first paper that I published with her back in 2007. That's how old I am. And what we found, again, at the time, we just looked at sort of what now known as the canonical myomeres, MIR1, 133, 206. And what I'll just have you focus on is the loss of MIR1 expression after 7 days of mechanical loading. A few years later, Thomas Chailloux joined the group, and this is some microarray data where we did 3, 5, and 7 days of mechanical overload. And you can see even after 3 days, there's a dramatic loss of miR1 in the muscle in response to mechanical loading. And I can tell you, I don't have it up here, but this seems to be independent of changes in primary microRNA expression. So something's happening. And for me, it was always very curious, how is miR1 being so dramatically downregulated within the muscle? So at the time, for us, it was around 2014 or so. Exosomes were just coming on board, and I was definitely inspired by this paper out of Russ Price's lab, led by Matt Hudson, in which they had an in vitro model of muscle atrophy where they treated myotubes with dexamethasone. And what they reported was that miR23, which represses atrogen, and MRF1, that miR23 was being exported from the myotubes via exosomes, and this was leading to this de-repression of these atrogens. And so definitely inspired by that, we thought, well, maybe in fact that's what's happening with miR1 in response to mechanical loading, that miR1 is being packaged into exosomes and being exported by the muscle. And this also made sense in this because miR1 targets a lot of pro-growth genes, so this would be a way for the muscle to, rapidly downregulate or remove that brake and enhance muscle growth. So this work was really directed by Yvonne Vichetti, who now he's an assistant professor at the University of Nebraska. And what Yvonne did, first we did a microarray analysis, and we went all the way down to just 1 day. And the first thing that I'll have you note is that miR1 is by far the most abundant microRNA, I'll have to qualify, in the mouse plantaris muscle. It's about 2.5-fold higher than any other microRNA. But even by a single day, you can see that it's dramatically downregulated. And so Yvonne confirmed that by qPCR, that you see about this over 50% loss. And again, even at day 1, that appears to be independent of primary microRNA 1 expression. So we isolated EVs. I'm not going to say how we did it, given the company, but we could find in these EVs that miR1 was more abundant after the first couple days, and then it returned to baseline in terms of the serum EVs. This is just us showing that somewhere in our EVs are, I'll say, exosomes that contain sort of classic markers of exosomes. But to be safe, I'll just refer to them as extracellular vesicles. Again, just for the sake of time, Yvonne went on to show that in response to mechanical loading induced by synergist ablation, these EVs contain miR1. And he actually found when he would isolate these EVs from either sedentary or, well, a sham or a mouse had been overloaded for a single day, label those EVs, reintroduce them into a naive mouse, we found that they were preferentially taken up by white adipose tissue. And then through microarray analysis, we found that, in fact, what was happening in fat that had received miR1, that we showed enhanced lipolysis. And that seemed to be directed by an increase in expression of adrenergic beta receptor 3. We showed that this appeared to be the mechanism by which you were getting increased expression. Actually, our in vivo data was stronger than our in vitro data. But regardless, when miR1 was delivered to white adipose tissue, it caused enhanced lipolysis. And that was probably because of an increase in epinephrine sensitivity or catecholamine sensitivity because of increased expression of adrenergic beta receptor 3. I should have. So it appears that Gretchen Reynolds likes exosomes. So we also contacted her based on this study that she, because the idea that muscle is communicating with fat and somehow that's being coordinated in response to resistance exercise. I don't know what Mark's experience was, but I went and I looked at all the comments of our paper when it was, our article was published in the New York Times, and I would say 90% were guys my age talking about their workout, and nothing about the science. So another aspect of this project is this mechanism conserved in humans? And so we call this our timer study. Basically, we have individuals, normal BMI is also high BMI. And what we do 60 minutes prior to just a really intense acute bout of resistance exercise, take blood, muscle biopsy, fat biopsy, do the exercise, then immediately collect blood. Again, at 30 minutes, take another biopsy and more blood, as well as another fat biopsy, and then collect blood at 60 and 90 minutes. And so here, again, this is actually a lot of people, but currently you and Wen and Lauren Deppa, who's an undergraduate, are really driving this project. And again, the numbers are relatively small. I'm sure like everybody, we've been impacted in terms of recruitment because of the COVID. But the trends are consistent with what we see in the mouse. I mean, I think for us it was somewhat surprising that MIR1 levels appear to be higher after 30 minutes as opposed to just immediately, which is, you know, EVs typically clear quite quickly after exercise. Within the muscle, except for these two guys, the trend is definitely either staying the same or there's a loss of MIR1 expression. And then within white adipose tissue, again, there's a lot of variability, so we'll just have to wait and see once we get the numbers. But I think at this point we feel like it's mildly consistent with what we see in the mouse. Also, we have the ExoView R100 platform, and this has already been touched on, but this is a really powerful technology in a way it allows us to begin to really look at the different subpopulations of EVs in terms of the classic tetraspanin markers, CD81, CD9, and CD63. And just to give you a little bit of flavor of what we're doing, and again, this is in the human, but we also can do the same analysis as the mouse chip that we're using. And this is just for the CD63 subpopulation. And you can see here, again, we're looking at between normal participants with normal BMI and ones with high BMI. And this is all relative, you know, set to 100%. So the first thing you can see is that just EVs, or in this case, they are actual exosomes. The exosomes just with CD63 are the most minor, whereas CD63, CD9 appear to be the most abundant. In response to exercise, you can see while it's just trending, it appears that in individuals with normal BMI, we see an increase in the abundance of CD63, CD81 exosomes, whereas it's unchanged in individuals with high BMI. And then here, where those EVs, and these again are some of the more abundant, but CD63, CD81, CD9 exosomes, here under resting conditions, it appears that individuals with a high BMI they're more abundant in those individuals. So I think this technology is really exciting because it will allow us to start to define these different subpopulations, how they respond to exercise or what form of exercise. So we kind of anticipate that hopefully, like in response to mechanical loading, you'll see differences in these subpopulations as opposed to we use a model of progressive weighted wheel running. Okay, so kind of returning back to MIR1, again, I have to give credit to Kevin. Kind of embarrassed about this. So as you know, I've been interested in MIR1 since 2007. I've always wanted to have a floxed MIR1 mouse. There are different versions out there where people have floxed both MIR1 and 133, but always just wanted one where both alleles of MIR1-1 and 1-2 were just individually floxed. somehow Kevin came to me and said, look at this paper. I was like, oh my gosh, we could knock out MIR1 specifically in skeletal muscle, repeat our experiment in terms of communication with white fat, what happens when you knock out MIR1 just under resting conditions, is it sufficient for muscle growth? So we went in on it together and re-derived the mouse, crossed it to our HSA mercury MER mouse. I lost a bet on this one. I was really hoping that when you knocked out MIR1 in skeletal muscle, that would look like to the muscle that it had been mechanically overloaded and it would be sufficient to induce growth. But as you can clearly see here, this is after a month post-tamoxifen. There is no difference in the weight of the lower hind limb musculature. We have studies where now we're taking the knockout MIR1 mice and subjecting them to either mechanical load or this progressive weighted wheel running, so see if maybe we can enhance the growth response. However, again, somewhat embarrassed because this paper again came out in 2014. Must have been a blind side for me, but this is an absolutely remarkable paper because they used C2C12s, so take that for what it's worth, but they found in C2C12s myotubes over 40% of MIR1 was found within mitochondria. And it actually, MIR1, it actually enhances the translation of mitochondrial-derived transcripts. It's very interesting. This paper I found absolutely fascinating. A little nervous because there's no follow-up on it, but we basically said, well, what's happening, where is MIR1 in adult skeletal muscle? So this work was done by Ahmed. This is just showing you, and again, this is just, here's just one, but I mean, we've done a lot of mice, but after about a month post-tamoxifen, we get about a 60% decrease in MIR1, which is not, it's not great, it's okay, but you know, our experience with trying to knock down microRNAs in muscle, we have a paper where we published a dicer knockout, muscle-specific dicer knockout, and even in the absence of dicer, we were unable to deplete microRNAs by more than 50%, and this is after 2 years. So there is something really unusual about microRNA stability in skeletal muscle. I really don't know what it is, but anyway, so 60% is okay. However, he fractionated to cytoplasm in mitochondria, and here he just normalized to the cytoplasm, and so the amount of MIR1 is about 29-fold higher within the mitochondria. Now, I put here just the CTs because the way in which it was normalized, I think, was driving some of that large fold difference, but even here you can see, just based on CT, that the abundance of MIR1 in the mitochondria was 1.5 CTs higher. So bottom line, there's definitely MIR1 within mitochondria. Now, you go back to this, and I was happy to see that the other speakers were touching on this, right? So I told you our story about how in response to mechanical loading, muscle releases EVs that contain MIR1, and that it appears that they preferentially are taken up by fat. So does that mean what we also may have been looking at was that mitochondria was being, in response to mechanical loading, mitochondria was being exported? And this was all new to me 6 weeks ago. But in fact, there are mitochondrial-derived vesicles involved in muscle adaptation and regeneration. There is other work in which people have shown that these mitochondrial-derived vesicles can be involved in mitophagy. So I think we're really excited about the possibility that what we're seeing is that in response to mechanical loading, we are exporting aspects of mitochondria. So I am not a mitochondrial expert, but by looking at the posters, I'm in the right place to get some ideas about really how to really make sure that our first look at MIR1 in mitochondria, that we're doing it in a rigorous way to confirm it, and then actually what is it doing in what is MIR1 doing in mitochondria? Okay, so I'll just finish with what I consider to be probably provocative summary points, but from our, and this is again all based a lot on our unpublished data, but this is where we're heading. So it does appear that some satellite cells appear to be actively communicating to the myofiber under resting conditions. So that adds a new dimension, right? So we've always been looking at when satellite cells become activated and then they start to communicate, but it looks like even under resting conditions, satellite cells may be communicating to the myofiber. Of course, and that asks the question, what is that communication? And again, the other motivation for us to develop tissue-specific ways to label exosomes is so that, one, we can try to track where they're going, but also the ability to be able to isolate them and characterize their cargo, and how does their cargo change in response to different types of exercise, how does it change with aging? Also, again, I think what's going to be happening over the next few years is that we're going to have the ability to really define these subpopulations of exosomes, and how do they change in response to exercise or different forms of exercise. How do they, one thing that's a big open question is the targeting of exosomes to different tissues, and is that related to these different subpopulations? And then finally, probably the most provocative and really early, but we're very excited by it, so I wanted to share it with everybody. Is MIR1 a mitomere? And if so, what is MIR1 doing in mitochondria? And are mitochondria being exported in response to a hypertrophic stimulus? And what role does that play in the hypertrophic growth? Okay, so with that, of course, a lot of people to acknowledge our funding, as well as the Center of Muscle Biology and our colleagues Esther and Chris Fry. Thrilled that Chris, we got him to come back to UK. But again, right now, Ewan Wen and Ahmed, I have a graduate student, Jensen, that are really driving it. And then, of course, all former members of our group. And with that, I'd be happy to take any questions.
Certainly very provocative talk. I think in the interest of time, we're a little late, so if anyone wants to have a chat with Dr. McCarthy, perhaps we could do so after coffee. But I'd like to thank everyone for their attention. I think what we realize, this is a really emerging area, so I hope the grad students and postdocs take this on, and we learn lots more in the next 10 to 15 years. Thank you.